So, in the next two chapters, 14 and 15, we're going to talk about host defenses. In chapter 14, we're going to talk about the innate defense systems of the body, and in chapter 15, we're going to talk about the specific immune response. So in chapter 14, it actually talks about your media under the microscope about tattoos. And I just think this is a really, really cool tattoo. And you can thank, however, one of the cells we're going to talk about here in this chapter, and that is our macrophages, because they are the ones that are going to take up the ink particles and actually keep the tattoo. So, host defenses. We have three lines of defense. The first lines of defense are barriers that block invasion at portals of entry, and they do not involve recognition of foreign substances, and we are born being able to do them. So they are what we call innate, and they are general in their action. Our second line of defense is also nonspecific and innate, and it is an internalized system of protective cells and the process of inflammation and phagocytosis. And so in this chapter, we're going to talk about these first two lines of defense. But I'll mention the third line of defense in preparation for the next chapter, and that is our acquired or specific immune response. And this is acquired after birth on an individual basis as each foreign substance is encountered by our lymphocytes, and we mount a specific response to that specific antigen. While the inflammatory process, phagocytosis, and the acquired immune response all work hand in hand, there are some differences in all of them together. Reaction of each microbe in the third line of defense produces a unique protective substances and cells that can come into play if the microbe is encountered again. One of the big differences between inflammation and the adaptive or acquired response is the adaptive and acquired response creates memory. So the adaptive or acquired response produces long-term immunity, whereas inflammation is a rapid onset, but usually doesn't last very long. So we can actually again divide our host defenses into the innate and nonspecific and the acquired or adaptive or specific responses. Under the innate responses, our first lines of defense are those physical and biochemical barriers, especially at the portals of entry. Our second line of defense is really inflammation, but we'll talk about phagocytosis, antimicrobial proteins, and systemic fever as a part of that inflammatory process. And then the third line, the acquired or adaptive immunity, this includes specific host defenses that must be developed uniquely for each microbe that the person comes in contact with. And this is under control of our B and T lymphocytes. There is some overlap between the second and third lines of defense, and we'll talk about those when we get there. So let's look at the first lines, our barriers. And so just think about these. First of all, I said intact skin. Not very many microbes are going to get through that. Also, mucous membranes. Not only are they stratified squamous epithelium, but they have all that wonderful mucus. What about pH? This is one of our biochemical barriers. What about just sloughing off of skin? So we'll look at some of these as we go through here. So our skin, as long as the skin is intact, it is very hard to get through. The epithelials are compacted, they're cemented together with tight junctions and desmosomes and impregnated with that waterproofing protein called keratin. It provides a thick, tough outer layer that is highly impervious and waterproof. Very few pathogens can penetrate this unbroken layer. Hair follicles. The hair shaft periodically is extruded, just like the epithelial surface cells. And so the follicle cells are actually disquaminated, they're sloughed off, and the hair is actually sloughed. Sweat glands, the flushing effects of the sweat coming out of the sweat ducts actually helps remove microbes from the surface of the skin and within the glands themselves. Mucous membranes of the digestive, urinary, respiratory tract, and even the eye and potentially the ear are typically moist and permeable, even though they're made up of stratified squamous epithelium. Mucus coat impedes the entry and attachment of most of our bacteria, and the blinking and tear production rids the eye of potential pathogenic irritants. The flow of saliva will carry microbes to the harsh pH condition of the stomach. In the respiratory tract, the nasal hairs within the sinuses will trap large particles. Copious flows of mucus and other fluids during allergies or colds actually exert what we call a flushing action. And in fact, this flushing action of any body secretion is called mechanical clearance from the body. The respiratory tree of the tracheobronchial tree is lined by ciliated pseudoepithelium that moves foreign particles trapped in the mucus layer towards the pharynx, where we either swallow them or we spit them out. So here you can actually see the cilia that are lining the surface of the epithelial cells lining the trachea and the primary bronchi. The genitourinary tract, this protection here is primarily through the continuous trickle of urine through the ureters and the urethra and the flushing of the bladder emptying. Vaginal secretions provide cleansing of the lower reproductive tract in the females. And certainly resident microbiota on the surface of the skin provides that microbial antagonism that we talked about previously. They help block access of pathogens to the epithelial surface so they cannot adhere and get established. They can also create unfavorable environments for pathogens by actually creating antimicrobial peptides of their own. So the normal microbiota competes for both nutrients and space with the pathogens that try to move in here. The resident microbiota can also alter the local pH if an attack is underway. So the skin and mucous membranes, the sebaceous secretions, the oil secretions exert an antimicrobial effect. Most microbes do not like fat, and so they tend to not actually cause as much disease if you have more oils on the surface of the skin. Also, in some body secretions, we have something known as the lysozyme. This is found in high amounts in saliva, tears, and even mucus. It hydrolyzes the peptidoglycan in the cell walls of gram-positive bacteria. So think about it. You get a paper cut. What is sometimes the first thing that you do? You stick that finger in your mouth. There's a reason for that. That saliva contains these lysozymes that will actually start to break down the gram-positive bacteria. Lactic acid and electrical or electrolyte concentrations in sweat, so the salt or the acidic conditions that can be found in various body secretions are also going to be part of these nonspecific chemical defenses. The skin's pH is actually somewhere between about 3 to 5. So again, most of our pathogens are more neutrophilic, and so they don't want it to be any type of acid. The stomach acid, made up of hydrochloric acid and gastric secretions. Normal pH is about 1.5 to 2. And I have actually seen this happen that one of my patients swallowed a penny and we got it out later on, and this is what it looked like. The acid had actually eroded away the copper coating on the penny and actually had begun to break up the zinc slug. The intestines, the digestive enzymes can oftentimes break down these microbes. Bile actually creates a slightly alkaline pH, especially in the upper part of the small intestine, to help neutralize the acid coming from the stomach. And then semen has antimicrobial chemicals added to it. And the vagina is a very acidic environment that's maintained by the normal microbiota. Now, there is a genetic variance in hosts. Some hosts are unaffected by infectious diseases that can affect other hosts. Some of this just simply be because of the the genetics that you have, in the fact that you don't produce something on your cells that the microbe needs to attach, where somebody else would actually produce that in their genetics. Some pathogens have such specificities for one host that they cannot actually infect another host. And this is sometimes those diseases that are only seen in one specific species of animal. So I often get the question about parvovirus in canines. Parvovirus is a viral disease of the intestinal tract in canids. And I oftentimes get the question, can my child get parvovirus? And the answer is no. Canid gut epithelial cells actually have a receptor for the spikes of the parvovirus. We do not have that receptor, and therefore, even if we did get the canine parvovirus in our digestive tract, it would simply pass through and not be able to adhere. So viruses oftentimes will only infect by attaching to a specific host receptor, and if you do not have that host cell receptor, the virus can attach. And this can also be true for certain bacteria. Those with a loss or lack of complete immune system are certainly more susceptible to infection. And if you've never had a chance to actually look up any information about the boy in the bubble, and I'm not talking about the movie, I'm talking about the actual young man who was born without an immune system. And he lived basically his very short life of 12 years in a sterile environment. But we can also see victims of severe burns because we've lost that epithelial barrier and microbes can just get in very easily. Blockages of tear ducts or salivary glands or even the intestinal or urinary tract can cause problems with microbes being able to exit the body. The first lines of defense alone, therefore, is not sufficient enough to protect against infection. Many pathogens have found ways to circumvent these barriers. So, the skin produces what to create a waterproof barrier to microbes? A thick layer of what is found in the nasal and respiratory passages that will trap bacteria and other microbes. Tears and saliva can contain something that would hydrolyze the peptidoglycan of the gram-positive cell wall. And the low pH in both the blank and the blank prevents the growth of microbes within those areas. And true or false, some pathogens are able to circumvent the physical and biochemical barriers of the first lines of defense. Now, immunology. Immunology is the study of the immune system. And really, that is looking at all of the immune system. But since the first lines of defense are really something that are either physical barrier or biochemical barriers, sometimes we don't really look at those very much. But they are actually part of the immune system. But really, immunology tends to focus on the second and third lines of defense. It is central to the study of the fields of both cancer and hypersensitivity reactions such as allergy or autoimmune diseases. A healthy functioning immune system is responsible for immune surveillance of the body. So our immune system is continually checking for foreign or abnormal substances in the body, recognition of any foreign or abnormal substances, and destruction of those entities that they find. So let's look at inflammation and phagocytosis, and this is the second lines of defense. White blood cells move throughout the body searching for potential pathogens, and there are a number of these. They are trained to recognize self and leave self alone, and or to recognize body cells and to recognize non-self cells that are either foreign or abnormal, and to get rid of those non-self cells. On our cells, we have groups of proteins known as tissue markers. Later on, we will find out that the actual name of these are the major histocompatibility antigens. These markers actually are like name tags on our cells that says to our immune system, this is me, please don't kill me today. These tissue markers are usually composed of proteins and or sugar molecules that literally are our name tags or our self-antigens. And so our immune surveillance system of the immune system will be evaluating these markers and deciding whether or not this is a self cell or a non-self cell. So when we come in contact with cells that are self, we go, okay, you guys are good. You're who you say you are. You can go on your way. But if we come in contact with a foreign or non-self cell, such as a bacterium here, we'll go, you're not right. And so oftentimes these cells will actually phagocytize whatever this non-self substance is and actually destroy it within the cell. And we'll look at that process. So describe the three responsibilities of a healthy immune system. True or false, cells marked as self are not marked for destruction by the immune system. And markers are composed of what biochemicals? True or false, non-self cells such as food or commensal organisms are attacked by the immune system. Now, the systems involved in the immune defenses. Body compartments that participate in the immune function include the mononuclear phagocytic system, the spaces of the tissue cells, and this is where the extracellular fluid is contained, and the bloodstream itself. And the lymphatic system is going to play a role in all of this as well. So, at the microscopic level, clusters of tissue cells are in direct contact with reticular cells and the extracellular fluid. Blood and lymphatic capillaries will also be present in this area. And cells and chemicals that originate in the reticular system or the extracellular fluid will diffuse or migrate into the blood or into the lymphatic capillaries. The flow of events depends on where an infectious agent or foreign substance first enters the body. So a typical progression would be extracellular spaces and reticular tissue. So let's say we get through the epithelial tissue and we get into the extracellular space of the connective tissues. We then can be picked up by the lymphatic circulation, which eventually takes that to the stream, and then we can deal with it. Now, what we'll find is this right here. So, for example, here's the extracellular fluid in between our cells of our organ or our tissue. We'll have blood vessels and we'll have lymphatic capillaries in here. Whatever these foreign substances would be will then come in contact with cells that are looking for them and actually will then be directed oftentimes either into the blood or into the lymphatic system, which eventually winds up in the blood as well. So what is the mononuclear phagocyte system? This is a supporting network of connective tissue fibers, and this is known as the reticular system. It originates in the basal lamina of the connective tissues. It interconnects nearby cells within tissues and meshes with the massive connective tissue network surrounding all organs. We also will find a whole bunch of lymphoid tissue throughout the body. We have GALT, which is gut-associated lymphoid tissue, and this is lymphoid tissue found in the lining wall of the intestinal tract. We'll have mucosa-associated lymphoid tissue, or MALT, and this can be found in the walls of any mucosal-lined organ or tube. And then we have some other specialized that I'll talk about a little bit later on. So within this mononuclear phagocytic system, we're going to have macrophages, we're going to have neutrophils, we're going to have a specialized macrophage called the dendritic cells. We'll have reticular fibers that are basically holding all of the parenchymal cells of the organ or tissue together. So the lymphatic system, along with these unorganized gut or associated lymphoid tissues, include the lymphatic circulation as well as the lymph nodes. And these provide an auxiliary route for the return of extracellular fluid to the bloodstream. The lymphatic capillaries actually act kind of as a drain-off system for the inflammatory response, therefore. They render surveillance, recognition, and protection against foreign materials through a system of lymphocytes, phagocytes, and antibodies. Now, lymph is a plasma-like fluid. It actually came from the plasma of the blood cell, enters into the extracellular space, and then will be picked up by the lymphatic capillaries. It's made up of primarily water, dissolved salts, and somewhere about 2 to 5% of proteins. It can transport numerous white blood cells, fats, cellular debris, infectious agents, and others that have gained access to the extracellular tissue space. Lymphatic vessels are found everywhere in the body. These are very thin-walled, more closely resembling the venules of the cardiovascular system than the arterioles. And they actually will then permeate and pick up extracellular fluid that has escaped from the cardiovascular system and has not been picked back up by the bloodstream. Two differences between blood and lymphatic vessels. One, lymph moves only in one direction, from the extremities towards the heart. There are no lymphatics going away from the heart towards the tissues. And lymph is only transported through the contraction of the skeletal muscle pumps. So, the lymphatic vessels will parallel the venous tree of the cardiovascular system. And so together, this whole, or both of these systems put together, are actually known together as the circulatory system. So lymphatics will only flow towards the heart. It actually will come back into the venous side going towards the heart. But again, there's no lymphatics going away from the heart as with the artery. So we only parallel the venous side of the cardiovascular system. Lymphoid organs and tissues are loose connective tissue frameworks that actually will house then lymphocytes, macrophages, and some other white blood cells. There are two primary lymphoid tissues, bone marrow, and the other one is the thymus gland. The thymus gland actually sits in the anterior part of the thoracic cavity, and we'll talk about it in a moment. Secondary lymphoid tissues include the organized lymph nodes that are associated with the lymphatic vessels, the spleen, which actually cleans both blood and lymph, and the secondary lymphoid tissues that are unorganized, such as, again, the MALT, the GALT, the skin-associated lymphoid tissue, breast-associated lymphoid tissue, and even the tonsils and adenoids. The thymus starts out life as a triangular structure that's fairly large just below the pharyngeal region in the upper portion of the thoracic cavity. It's largest at birth and will begin to atrophy or get smaller at puberty. It shrinks gradually through adulthood so that by the time you're in your 70s or so, pretty much you have no functional thymus gland left. This is one of the reasons why the specific immune response decreases as we age. It is the site of T lymphocyte cell maturation, and we'll talk about that in the next chapter. So, it starts out here as fairly large. It sits over the great vessels of the heart, and then at puberty actually begins to get smaller over time. The lymph nodes are these small encapsulated bean-shaped structures that run along the lymphatic capillaries. They can be found in small groups or larger aggregated groups along the lymphatic channels. Major aggregations are oftentimes named, such as the axillary lymph nodes found in the armpit, the inguinal nodes in the groin, the cervical nodes in the neck, or the mesenteric nodes scattered throughout the mesenteric tissue of the abdominal cavity. They're ideal for filtering out materials in the lymphatic fluid as it flows through the lymphatic circulation. The spleen is the largest lymphoid organ. It's located in the upper left quadrant of the abdominal cavity, and it actually filters primarily blood instead of lymph. But in the white matter of the spleen, we actually will see lymph being filtered. So the primary function of the spleen is actually to remove worn out red blood cells from circulation. It can also house platelets, but its immunological function includes filtering pathogen from the blood for phagocytosis by macrophages and lymphocytes in the white islands of the spleen. Miscellaneous lymphoid tissue is just bundles of lymphocyte, not really encapsulated, that lie beneath the mucosa of the GI and respiratory tracts primarily. So the tonsils would be one of these miscellaneous lymphoid tissues. We'll actually see BALT in the breast tissue and GALT, malt, and salt in the GI system, the mucosa, and the skin. Peyer's patches are actually somewhat organized groups of lymphocytes and macrophages found in the lining of the small intestine. Now, how does the blood function in the immune system? Well, just a quick review. Whole blood is composed of the blood cells suspended in the plasma, which is that clear yellowish fluid primarily made up of water. Serum is simply plasma, except that the clotting factors have been removed. So the blood that has clotted, what the clear portion will be serum. And oftentimes serum is used in immune testing and therapy. So when we actually take a red tube that it got anticoagulant in it and spin it down, the red cells and the white cells will go to the bottom and the clear portion will be plasma. However, if this was a clot tube that did not have an anticoagulant in it, what we would actually be seeing is that this would be forming a clot, and then we would have serum up here. Some components of the plasma, it's mostly water, and then we have some proteins, mostly albumin with some globular proteins or globulins, and this does include the antibodies, and then fibrinogen, which is the inactive form of fibrin used in the blood clotting mechanism. We also will have some hormones dissolved, some nutrients certainly, certain ions, and oxygen and CO2 as dissolved gases, along with some nitrogen. And then we also will have waste products that are usually headed towards the kidney or somewhere else to be excreted from the body. Hematopoiesis is the making of the blood cells in the bone marrow. Early on during embryonic development, the blood cells are first made by the yolk. And yes, we started out with a yolk. As the liver actually begins to get large enough, that production of red cell or of blood cells is taken over by the liver and the developing lymphatic system. And eventually, once enough of the red bone marrow has been produced, then it takes over completely in making the blood cells. So early on, it's the yolk sac making our blood cells, then the liver, and then finally this red bone marrow found within the bones. Now, stem cells, we have what we call a hemopoietic stem cell, and this is the precursor to all of the blood cells. This is a pool of undifferentiated cells. So these hemopoietic stem cells can actually become a red blood cell, white blood cells, or even a platelet. These are maintained in the red bone marrow and then will develop under specialized instructions. So the hemopoietic stem cell can actually become a stem cell for erythroblasts that become the erythrocytes, the megakaryoblast, which become the platelets, or myeloblasts, which become our granulocytic white blood cells, the monoblast becoming the monocyte that becomes the macrophages, dendrites, and other specialized macrophage cells, and the lymphoid stem cell, which will give rise to our B lymphocytes, T lymphocytes, and our natural killer cells. The leukocytes can be evaluated by reactions to hematological stains that contain a mixture of both acid and basic dyes. So cells can be differentiated by color and morphology. They appear with or without colored granules in the cytoplasm. The granulocytes have these stained granules in the cytoplasm as well as lobed nuclei. The agranulocytes are unlobed and they have rounded nuclei. The granulocytes, if you remember, are the neutrophils, the eosinophils, and the basophils. They all contain cytoplasmic granules, with the neutrophils staining somewhat neutral, eosinophils staining reddish, and the basophils staining very blue. The granules function in numerous physiological events within these cells. So just to remember, there's our good old neutrophil. It is the most common white blood cell that we see in circulation, and there's that lobulated nucleus. Here's the eosinophil, and I don't really like this picture real well because really this should be kind of red in these granules. And then the basophil, when you see one, you know it because the big blue staining granules are just all in that. Really, to me, this one almost looks more like a eosinophil than a basophil. Neutrophils make up around 60 to 70% on average of circulating leukocytes. They produce toxic chemicals and are phagocytic in function. Eosinophils, the granules contain peroxidases, lysozymes, and other digestive enzymes, along with toxic proteins and inflammatory chemicals. And they can attack and destroy large eukaryotic pathogens such as our worms and flukes. The basophils are the least numerous in circulating white blood cells. They share the morphology and functional characteristics of mast cells that are found within all connective tissues except for the blood. And so we believe that actually the basophils are going to be the cells that are the driving cells of inflammation within the blood itself. The agranulocytes consist of two non-lobulated nuclei containing cells that lack the predominant cytoplasmic granules. And we have two types, the lymphocytes, which have a very large dark staining round nucleus, and the monocytes, which are the largest in size of the circulating white blood cells. The monocytes will actually become the tissue phagocytes. So the lymphocyte, not much bigger usually than the red blood cell, but a big blue staining nucleus with a little ring of cytoplasm around it. The monocyte is very large, usually about 2 to almost 3 times the red cell. They usually have kind of a kidney bean shaped nucleus. This one's not really showing it real well, but kind of the kidney bean roundish type nucleus that we can see. Once the monocyte leaves the bloodstream, it's then activated to the macrophage. The lymphocytes comprise about a third of all circulating white blood cells at any given time, and it's estimated that about 1/10 of all adult body cells are actually lymphocytes. They exceed only, or they are exceeded only by red blood cells and fibroblasts. Three functional types of lymphocytes. We have the B lymphocytes, and I'll talk about the bursal equivalent in the next chapter. The T lymphocytes that are thymus derived, and something known as the null or natural killer cells. Activation of the B lymphocytes, these are going to be in charge of what's known as humoral or antibody mediated immunity. They will form plasma cells that produce a large protein called an antibody. And these antibodies will interlock with the antigen and participate in their destruction. And again, we're going to talk about this in chapter 15. T lymphocytes are in charge of cell-mediated immunity, and there's a wide spectrum of immune functions. We have several subclasses of the T lymphocytes, and again, we'll talk about those in the next chapter. They modulate immune function and will kill foreign and abnormal cells. Both B and T cells account for recognition and the memory that we see in the adaptive response. Monocytes, again, the largest, they make up about 5% of all white cells in circulation. And the reason for that is actually they are going to be exiting and going out into the tissues fairly quickly. The cytoplasm of monocyte does have some very small granules that contain digestive enzymes, but these are not easily seen upon staining. They are discharged by the bone marrow into the bloodstream and travel to the tissues where they are needed, exit the bloodstream through a process known as diapedesis, and actually then become the macrophage. The macrophage is very long-lived in the tissue and actually does have the ability to undergo mitosis. They are among the most versatile and important of all of the white blood cells in our immune system. Functions of macrophage, they have both specific and nonspecific killing functions. They actually are in charge of cellular housekeeping. They phagocytize messes created by inflammation, infection, dead and dying cells. They process foreign molecules and present them to lymphocytes to actually activate the adaptive response. They secrete biologically active compounds that assist, mediate, attract, and inhibit immune cells and reactions. And some of them actually are named for their various specific functions. And a good example of this is the dendritic cell. The dendritic cell is a modified macrophage found primarily in the lower parts of epithelial tissues and in connective tissues. They move from the blood to the MPS and the lymphatic tissue, so really out into the environment of the body, where they then trap pathogens and engulf them and then process the antigens. So they ingest these bacteria and viruses and stimulate the cells then to move from their location to lymph nodes or the spleen. They mature into highly effective processors and presenters of foreign proteins to other cells of the immune response. And this is just a picture of one of these dendritic cells. And all of these cytoplasmic extensions, and they're not flagella or cilia, but they are just simply extensions of the cytoplasm and the cell membrane. They're out there feeling for anything. And if they come in contact with a cell, they're going, are you a good cell or a bad cell? And if you're a bad cell, it will actually trigger endocytosis or phagocytosis. And then the dendritic cell will actually detach from where it is and travel and tell other immune cells, this is the guy that's trying to get into the body. The erythrocytes, the red blood cells, of course, carrying our gases of oxygen and CO2, they don't have an immune function except for that they bring in oxygen for repair. The platelets do have a role in the fact that they are going to be involved in our hemostasis, our stopping of bleeding from damaged blood cells during inflammation. They are a cell fragment of the megakaryoblast, and so they become sticky in areas where cells or where blood vessels are damaged and actually form our platelet plug. And upon the platelet plug, we can then build our fibrin clot. So, describe the mononuclear phagocytic system. True or false? Lymph is propelled through the body by the actions of smooth muscle. What processes worn out red blood cells and filters pathogens from the blood? And describe the function of the following white blood cells. Now, phagocytosis. The general activity of phagocytes is to survey the tissue compartments, discover microbes, particulate matter, and injured or dead cells, and engulf them. They do this in order to eliminate these materials. And they must be able to recognize immunogenic information, and that is something we'll call the antigens of foreign material. Neutrophils are the earliest phagocytic cell that responds to injury. So they react very early in the inflammatory response. They are the first ones there. They come in huge numbers because they simply are in huge numbers in circulation, but they die pretty quickly. And so the neutrophils are the first phagocytic cell into an area of injury and or infection. Common sign of bacterial infection is oftentimes a high neutrophil count in the blood cell. Because they're dying off so quickly, we're going to send chemical messengers to the bone marrow, and the bone marrow is going to start chugging out more neutrophils into circulation. The eosinophils are attracted to sites of parasite infestation. And so we can actually see an increase in the eosinophils and eosinophilia, oftentimes with either parasitic infection or in allergies. They play a minor phagocytic role in antigen-antibody reactions. The monocytes transformed into the macrophages in the tissues will increase in size in the tissue. They have enhanced development of lysosomes and other organelles within the cytoplasm. Histiocytes are something we call fixed macrophages. These are macrophages that have migrated to certain tissues and tend to remain there during their long lifespan. So, the alveolar macrophages found in the alveoli of the lung. The Kupffer cells lining the sinusoids within the liver. And we could even say dendritic cells in the skin are a form of a histiocyte. But we find macrophages basically everywhere. The microcytic cells of the central nervous system are actually a specialized histiocyte that will engulf any foreign material that enters into the brain tissue. So here's one of these alveolar macrophages. Here in the liver cells, we have these Kupffer cells in the sinusoid capillaries that are going to be taking anything out of blood that's moving through those capillaries. Here's the Langerhans dendritic cells in the lower part of the epidermis. And these monocytes simply will emerge through the wall of the blood vessel, and we'll talk about that in a moment, and then become the macrophage out in the tissue. When a macrophage recognizes a foreign substance, this will actually bind to specialized receptors on the cell surface, and this will initiate phagocytosis. The product then is whatever we're bringing in will then be in this vesicle known as a phagosome. Lysosomes that contain the digestive enzymes will then move over and actually blend with the phagosome. And we call this whole thing then the phagolysosome. The digestive enzymes then will work upon whatever this foreign substance is destroying it, And eventually the macrophage will have this bag of trash, if you will, and this bag of trash then will undergo exocytosis so that this residual particles then will simply be thrown out of the cell. So this purple cell is actually a macrophage coming in contact with these bacteria and beginning to engulf them. Now, many organisms, microorganisms, actually have something called pathogen-associated molecular patterns, or PAMPs. These are recognized by phagocytes and other defensive cells. So these are usually complements of protein or other products of the cell wall of bacteria, the spikes of virus, the cell wall of fungi. And they serve as a signal molecule on the surface of the microbe. They are not present in mammalian cells. So some examples of these PAMPs include peptidoglycan, the lipopolysaccharides found in the gram-negative outer cell membrane, and double-stranded RNA found in certain viruses. On our phagocytic cells, oftentimes, we then have pattern recognition receptors that will then recognize these PAMPs. And they will bind to the PAMP, and that will initiate then endocytosis and phagocytosis of this foreign substance. Now, the pattern recognition receptors are found on the surface at all times, regardless of whether they've encountered one of these PAMPs or not. We also see something called toll-like receptors, and this is a type of these pattern recognition receptors that recognizes certain PAMPs of microorganisms. The toll-like receptors will set in motion a cascade of events inside the cell, the phagocytic cell, that will amplify and orchestrate a defensive response to the pathogen. And so this is a toll-like receptor, and here it comes in contact with the foreign molecule, and this contact with the toll-like receptor will cause certain genes to be read, and this cell will now not only phagocytize whatever this is, but will release a number of chemical mediators. Cytokines, which literally the word just means cell movers. They are cellular communicating chemicals between different cells of the immune response. Interleukins, which do very similar things, and a number of inflammatory mediators that will communicate between all of the inflammatory cells. Now, once the lysosome will migrate to the phagosome and fuse with it, again, we refer to that as the phagolysosomes. The digestive granules of the lysosome then will actually break down the ingested material. This causes death of bacteria within the phagolysosome within about 30 minutes after phagocytosis has occurred. Two systems of destructive chemicals are found in phagocytic cells. An oxygen-dependent system, which is actually going to use certain, again, free radicals of oxygen, and one of these is myeloperoxidase, which forms halogen ions, or OCl negative ions, and other oxygen products such as superoxide anion, singlet oxygen, hydroxyl free radicals. The other is going to be the release of lytic chemicals such as lactic acid, the lysosome, or nitrous oxide. Some of these cationic proteins, so positively charged proteins, and proteolytics and hydrolytic enzymes will also be produced. Four classic signs of inflammation. And so let's talk about the inflammatory response. So phagocytosis is really part of the inflammatory response. And inflammation is a nonspecific, rapid response to injury in vascularized tissue. And that's important. We have to break or damage blood vessels. So there's four classic signs and symptoms of the inflammatory response. Rubor, or erythema, which is redness. Calor, which is a localized heat. and tumor, not tumor, but tumor, which is localized edema. All three of these are going to actually be due to the fact that we are going to get vasodilation in these injured tissues and get an increase of blood into this area. And then the fourth classical sign symptom is dolor or pain. And pain is a good thing. Pain tells you that you have breached a first line of defense. We also have kind of a fifth sign of inflammation, and that is loss of function of the injured part. And this can be temporary, and usually is, or sometimes it actually can be permanent. All the signs and symptoms of inflammation serve as a warning that an injury and sometimes infection has taken place. Inflammation will set in motion responses that hopefully save the body from further injury. So, can you see the signs of inflammation here? And I'm not sure what's causing this inflammation. It might be due to an infection. It may just be due to something else. Not sure. Okay. Now, the localized heat is going to slow down the metabolism of the uh, foreign or foreign substances, but actually increase metabolism of the inflammatory cells. The chief functions of inflammation include mobilizing and attracting immune components to the site of injury, to set in motion mechanisms to repair the tissue damage and localize and clear away harmful substance, and destroy any foreign microbes and block their further invasion. And so we really can put all this into this. The primary goal of inflammation is to heal the wound. We have to do all of this in order to heal. And I always say we have to clean it up in order to heal it up. And that's what inflammation is going to do for it. So here we've had a splinter that has pierced the epidermal tissues. And we'll have bacteria on the splinter, bacteria that will enter the wound from the skin, and we've broken a blood vessel. Our immediate vascular response of the broken blood vessel is actually vasoconstriction. Those blood vessels, small blood vessels, are going to constrict down to stop the loss of blood through the injured area. However, that's not going to last very long. The next thing, or almost the immediate thing that's going to occur as well, is in the connective tissues we have these mast cells. Mast cells are the driving cells of the inflammatory response. When we see injury, what we're going to see is that the mast cells are going to release and make a number of chemical inflammatory mediators that are going to do several things. We're going to have neutrophils in this area immediately because we're bleeding. So the neutrophils are simply going to come out the hole. We're then going to get the platelet plug and blood clotting occurring to stop the blood loss. But we're going to continue this inflammatory process as time goes by. So within about 30 to maybe 40 seconds, our blood clot is working, and our blood vessels now, under the influence of some of these chemical mediators from the mast cell, now undergo vasodilation. This is going to then cause an increased blood flow into this area, and that's going to result in the redness or erythema, the increase of temperature, and the edema eventually. We will see that we then form this edema, which may actually then become an exudate. And lots of neutrophils early on. Macrophages wind up coming in a little bit later. I refer to the neutrophils as the Marines of this immune response because they're the first ones there, and a lot of them die in the process. They're real good about phagocytizing these foreign bacteria, but they're not real good about taking out the trash once the phagolysosome has been processed. So they fill up with all these residual bodies and it actually dies. The macrophages usually come in a little bit later and finally clean up the entire mess. Neutrophil numbers peak within about the first 24 hours after injury has occurred. Macrophage numbers won't peak until sometime around 3 to 5 days after injury has occurred. The blood clot will actually dry and form a scab, and that actually is going to form a temporary barrier for anything else getting into the connective tissue at this point. And then we'll have cells that will heal. We'll have fibroblasts involved. The endothelial cells of the blood vessels will undergo mitosis to replace the cells we lost in the injury. The epithelial cells here of the epidermis will regenerate, and we will form scar tissue, and eventually the scab will slough off, and what we're left then is with an area that has some scar tissue in the connective tissue and will be healed. So the earliest change in the vasculature in inflammation is going to be in the small blood vessels, the arterioles, capillaries, and to some extent the venules. The immediate response, I mean instantaneously, is going to be vasoconstriction to restrict blood loss through the defect in the blood vessel wall. As soon as the mast cells undergo their degranulation process and release the chemical mediators, we then will see these blood vessels begin to dilate. And what we'll then see is then the signs of inflammation. Chemotaxic factors or chemokines will actually attract neutrophils and macrophages to this area. By the time that we start to see the vasodilation, we should have at least the platelet plug intact in the defect in the blood vessel wall, if not the actual beginning of the fibrin clot. So we'll stop the blood loss and we vasodilate. The inflammatory mediators, some of them will cause systemic fever, some will stimulate lymphocytes, some of them can actually prevent viral spread if this is a virus. And some actually will cause hypersensitivity reactions or an overproduction of the immune response, unfortunately. So some of the mediators, and I'm not going to go through the whole list, but I want you to know a few of these. First of all, what we'll see is from the mast cell, they will release a very potent vasodilator called histamine. Histamine is a vasodilator and will cause the blood vessels as they dilate to become leaky. And so we say histamine also increases vascular permeability. And this leads to leakage of fluid from the blood, not the blood itself, but water and parts of the plasma into the extracellular space. This becomes the edema and eventually exudate. It also will release serotonin, which will actually assist in serotonin release from platelets and other cells. And this actually will inhibit gastric secretion, but is also by itself a vasodilator. We will also see interferons and interleukins and prostaglandins being released from a number of different cells. The interleukins have a variety of effects, including causing systemic fever. Some are vasodilators, some are vasoconstrictors, actually. Prostaglandins are also vasodilators, but some of them actually stimulate the nociceptors or pain receptors, and this is some of what can cause the pain. Mast cells will also release platelet activating factors to actually activate the platelets and help with the platelet plug. Now, edema. As these blood vessels get larger, the endothelial cells begin to pull away from each other, making them leaky. And so fluid as well as even small proteins will be able to exit from the plasma into the extracellular space. This causes the edema, the swelling, and eventually the exudate. And there's various forms of exudate depending upon whether this is just injury that's caused inflammation alone or inflammation with the infection. The edema is a localized swelling and firmness due to accumulation of the exudate within the extracellular space. This outward flow of fluid from the blood will also assist in getting the white blood cells through the wall of the now fixed blood vessels, and this is known primarily as diapedesis. This is the movement of cells from the blood through the blood vessel wall into the tissue space. And this occurs because white blood cells are actively motile and are able to change shape and actually squeeze through, if you will, the endothelial cells of the capillaries. Receptors on the endothelial cells will help capture the white blood cells on the inside wall of the blood vessel and will actually participate in moving them through the wall of the capillary. So this is what happens. So we literally become kind of leaky. We still have the basement membrane intact, but we're going to get fluid coming out here in exudate. And what we'll see is as the blood vessel gets larger, blood flow slows down. And so these large nucleated cells, in comparison to the red cells anyway, will actually marginate. And so they kind of go to the edge of the blood column. They will stick to receptors on the endothelial cells. This will keep it from being picked back up by the bloodstream and float off again. And then the endothelial cells of the blood vessel will help guide them through the endothelial cell wall so that they wind up coming out on the other side. And these are actually some white blood cells that are squeezing through and going over here to the extracellular space. Chemotaxis is where we release these chemicals that will then actually attract white blood cells to migrate to a specific area. Cells will swarm from the different body compartments to the site of infection and inflammation and remain there to perform their generalized and specialized functions. Now, what is the benefits of edema and chemotaxis? Well, the influx of the extra fluid into the extracellular space helps dilute any toxic substances that may be there being produced by the foreign entities. The fibrin clot will actually help trap microbes and prevent them from getting into the bloodstream and traveling any further. Neutrophils, which again are the first ones there, actively phagocytize and destroy bacteria, dead tissue, and any particulate matter. Neutrophils and are usually going to be dying pretty quickly, and so oftentimes if we have a massive amount of neutrophils, we actually will see the exudate become a pus. The pus exudate is a whitish mass of dead and dying neutrophils, liquefied cellular debris, dead and dying bacteria. Later in inflammation, we'll see attraction of monocytes, lymphocytes, and more fixed macrophages. Macrophages will clear pus, cellular debris, dead neutrophils, damaged tissue. So again, neutrophils tend to peak within about the first 24 hours. Monocytes begin showing up, becoming macrophages within a very short period of time. But their numbers don't usually peak until sometime about 3 to 5 days after injury has occurred. B lymphocytes will be attracted to this area in the adaptive response to produce antibodies that will interact with any foreign antigens, and T lymphocytes will kill any abnormal or foreign cell directly. Once we clear up all of the cellular debris, the dead and dying stuff, fibroblasts from the surrounding connective tissues begin to aggregate, lay down connective tissue fibers, and actually form scar tissue. Systemic fever is an adjunct to inflammation. It's abnormally elevated body temperature and is usually nearly always a universal symptom of infection. However, I will say this. More often than not, fever occurs with bacterial infections. It doesn't always occur with viral infections. It can also be associated with certain allergies, some cancers, and even some organic illnesses such as diabetes. Low-grade fevers is anything about 37.7 to 38.3 degrees, or in our vernacular, about 100 to 101 degrees Fahrenheit. Moderate, 102 to 103. High fevers, anything above 106, we are very, very, very concerned. We need to get that fever down, and we need to get it down now. The reason for that is above 108 degrees, we start denaturing proteins, and the first cells that find their proteins being denatured are neurons. Pyrogens are actually chemicals that reset the thermostat in the hypothalamus, causing fever. Exogenous pyrogens originate from outside the body, and often these are products of infectious agents such as viruses, bacteria, protozoans, or fungi. They can be endotoxins, they can be blood or blood products, it could actually be part of the vaccine or another injectable solution. Endogenous pyrogens originate inside the body, and we actually produce a number of these from our inflammatory and immune cells. They can be released by monocytes, neutrophils, macrophages, and mast cells during the process of inflammation. Interleukin-1 and tumor necrosis factor are among the two most well-documented endogenous pyrogens. Now, benefits of fever. Fever can actually inhibit multiplication of temperature-sensitive microorganisms. Remember, most pathogens like it at a more middle temperature. They're mesophiles. So this can impede also the nutrient of bacteria by reducing the availability of iron to certain bacteria. It will also increase metabolism and stimulate the immune reactive cells and actually stimulate the entire immune process. It's a difficult decision sometimes to whether or not to treat fever to suppress it. Some advocates say a slight to moderate fever in an otherwise healthy person should simply be allowed to run its course. You may feel kind of rotten, but it's actually probably helpful. Potential benefits are there and there are minimal side effects. But all medical experts agree. High and or prolonged fevers are risky and should be treated so that we bring the fever down. Fevers in those with cardiovascular disease, head trauma, seizures, respiratory ailments should also be treated. Antimicrobial proteins, there are a number of these, and again, I'm not going to talk about all of them, but interferon, I think I've mentioned interferon already in previous lectures. Interferons are small proteins that are produced naturally by certain white blood cells and all tissue cells if they are virally infected. Interferons are used in therapy against certain viral infections and have also shown some promise in actually treating certain cancers. They are involved in defenses against other microbes and in immune regulation and intercommunications. But the big thing about interferons is they interfere with viral replication in the host cell. So, if I've got a cell that's virally infected, what this will trigger is the synthesis of interferons by this virally infected cell. Now, this cell may eventually die because of the viral infection. But what happens is the release of that interferon can flow through the tissue fluid over to another host cell that's not yet infected. It will bind with specific receptors that do one of two things. One, the binding of the interferon with these receptors will activate something known as the antiviral protein genes. This will then produce a number of antiviral proteins into the cytoplasm of this cell. So if this cell should be invaded by this virus, these antiviral proteins actually block the replication of the virus once it's in its cell. So we stop viral replication. The other thing that we think interferon may do is these receptors that the interferon binds to may be the actual receptors that this virus may need to bind to get into the host cell. So if interferon is blocking that receptor, we actually get neutralization of the virus, and it can't get in its cell, can't get in the cell, can't reproduce. Interferon is not necessarily virus specific, so synthesis in response to one cell type that's been infected may actually protect against other viruses in other cells. We actually can produce interferon industrially as a treatment for certain viral infections, however. Complement system. This is will produce antimicrobial proteins, and this is actually a blood cascade system. So in the blood, we have the hemopoietic or blood clotting system with a platelet plug and clotting mechanism. Complement is also a protein cascade similar to the clotting mechanism, but it produces a number of what we call complement proteins that are active in inflammation and the adaptive response. Complement proteins will work together to destroy foreign substances. This cascade reaction, the sequential physiological enzymatic response, will actually then produce a number of these complement proteins. And it's called complement because it complements or assists inflammation. There are three ways in which we can start complement. The classical complement pathway is actually started by the interaction of antibodies with its antigen on microbial cells. The lectin pathway is activated by a group of small proteins found in the blood called mannins that are activated when certain organisms enter the body. And the alternate pathway is activated by substances found in the bacterial or fungal cell wall, or the virus capsid or envelope, or even on parasite surfaces. So we get down to a common pathway, and this is complement 3, which is cleaved into its active forms, and then in turn will activate a number of other factors, some of which will interact directly with cells, such as those that will form what we call the membrane attack complex that punches holes in membranes. Well, when we punch hole in cell membranes, the cell contents are going to leak out and that cell is going to die. Some of the other complements will coat foreign substances to increase the chance of phagocytosis. This is the process known as opsonization. And some actually just stimulate further mast cells to undergo degranulation and release more of the inflammatory chemical mediators. So again, classical pathway, it is actually due to antibody-antigen complexes that form. The lectin pathways is due to the presence of mannins, which are actually activated by mannose that's found in certain bacterial cell walls. And then the alternative is other products that are found in the cell walls of some of these microbes. Antimicrobial peptides are just short little proteins that are capable of inserting themselves into many bacterial membranes. They usually run about 12 to 50 amino acids in length. There are a number of these: bacteriocins, defensins, manganins, and protegrins, and there are probably more that we haven't even discovered yet. They are actually considered part of the innate immune system. They do have effects on other actions of nonspecific and specific immunity both. So these just simply insert themselves into, again, oftentimes the cell membrane, basically breaking it apart and causing lysis of the cell. Iron is actually required by humans and bacteria for enzyme and metabolic function to properly run. In fact, many bacteria actually require iron as part of their electron transport chains to make ATP. So if I can limit the amount of iron available to the bacteria, I can limit their growth and reproduction. Iron-binding proteins then keep iron bound tightly so that it is not available for microbial use. Hemoglobin, located in our red blood cells, is actually one of these iron-binding proteins that we could actually make and release, not inside the red cell, but to actually just bind up iron in the hemoglobin molecule in order to keep it away from the bacterial cells. Transferrin is found in blood and tissue fluids, and it's just always present, but it oftentimes will increase in amounts if bacteria are present. Lactoferrin is found in milk, blood, tears, saliva, and probably other body secretions. And ferritin is a type of iron-binding protein found in every cell. The siderophores are actually proteins capable of scavenging iron from iron-binding proteins. And so some of the bacteria actually have these and will try to take the iron away from these iron-binding proteins. These siderophores will actually bind the iron actually more tightly than the human proteins, and therefore can actually be taken up by the bacteria. So, can you list and describe the basic steps of phagocytosis? What are the 5 cardinal signs and symptoms of inflammation? Blank is the tendency of white blood cells to migrate in response to a specific chemical stimulus. True or false, fevers should always be treated. Interferon is produced in response to infections by what specifically? What are the 3 ways that complement proteins are activated? And the iron-binding protein found in blood normally is what? So in Chapter 15, we'll take inflammation and then move into the adaptive, the acquired, or specific response, and I typically will call it the adaptive response, even though it is also known oftentimes as the acquired. It's called specific because it is specific to each individual. Sorry, dog was barking, but that's the end of the chapter, so see you next time.